or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Thank you for joining me.
Pleasure to be here.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. I'm a professor of genetics at Stanford University School of Medicine. I direct the Center for Genomics and Personalized Medicine. Our specialty is big data. Back at a time, people used to study genes one at a time, and we came up with ways of studying them all at once. It used to be on yeast. And so I think that's kind of guided our research ever since, trying to look at problems in a more comprehensive sense.
Mm-hmm. So can you give us a sense for what is the breadth of questions your lab looks at, and what are some of just give us a basic sense of some of the tools and technologies you deploy?
Sure. Well, I think what drives us is that we think medicine is broken. That is, we practice sick care, not health care. And so we're trying to transform that in many different ways. As an example, when we go to a doctor's office, we typically measure very few things there, and some of it's of questionable value. And then we treat people based on population averages. What we're trying to do is get a much, much more complete picture of people's health, do a lot of it by monitoring at home, and then actually treat people based on their personal profiles and their trajectories, which may or may not be like a population average. A good example is, you've probably been told since you're little, your temperature when you put a thermometer in your mouth is 90.6. But if you actually look at the data that are out there, it's more like 97.7, according to a big study out of Stanford.
On average.
People, on average. But the more important point is there's a spread. There's another study that says 25th quartile is 94.6. The 75th quartile is 99.1.
Wow. So from 94.5 all the way up to 99 is where most people sit.
Yeah, that's correct. And so what that means, if your normal healthy baseline is 94.6, you go to a physician today, they measure at 98.6, they're going to tell you you're healthy, you're normal, everything's fine. But I guarantee if you're up 4 degrees over your baseline, you're not healthy. Something's
Right,
up.
right. And I suppose another thing there is that that number, that temperature is itself dynamic. It's going to be a function of, what you eat, for example.
It is to some extent, yeah. And you'll have a little bit of fluctuation on a circadian pattern. Yep.
Okay, and then so what kind of just give us a basic sense before we go into some detail on some of it, because I know that you do a lot. What sort of technologies are you using? You're more of a molecular lab. You get down to the molecular level.
Yeah, molecular and genetics. Although these days we're getting a lot into wearables and physiology. So basically, I guess what we did about 15 years ago, stemming off some work we used to do on studying biological systems, is we tried deep profiling on people with the idea of monitoring health. So the technologies we use will sequence people's genome to try and make predictions about their genetic risk. We'll draw blood and urine and stool to look at their microbiome, and we'll do very deep measurements based on RNA, their proteins, their metabolites, and such. We've invented some of those technologies that are used today. Something called RNA-seq is one of these methods that came out of our lab. It's a way of looking at all your RNA molecules. Anyway, we'll measure that out of blood. Some of like metabolomics and things, your metabolites will measure that out of urine. Your microbiome, you may or may not know, it's super important for your health. It makes lots of essential vitamins, breaks down your food. We'll measure that as well. And so one aspect is to take very, very deep data on people, and the other aspect is to do it over time. So we'll actually profile people while they're healthy, not while they're sick, see what a healthy profile looks like. We do it every 3 months and additional times if they get ill, and we'll profile them over time. And the idea is to see what is your healthy profile, how's it changing, what's the difference between, say, you and me? How are we the same? How are we different? Turns out we're quite different. Um, and in fact, this is why what we're doing we think is so important for health monitoring. Turns out that a sick you is still closer to a healthy you than it is to me, meaning
Mm.
sorry, the difference between people is greater than the difference between a healthy and a sick
I see.
profile.
So if we did just some of the profiling you're talking about, never mind what the details are at this moment. You looked at my profile and your profile, and then you looked at my profile again after, say, I got sick.
Hmm.
My healthy profile and my sick profile would probably be closer to each other than my healthy profile would be to your healthy profile.
Correct. Because we're so different from one another. And that's why it's important to know your healthy profile. So when you get ill, you can detect that
Mhm.
shift.
And I
Uh,
guess kind of what you're getting at, or what some of the subtext is here, based on what you said initially, is when the way that we do medicine today, in essence, people are only going in and getting a partial sick profile after something goes wrong.
Correct. And so it's crazy. And then you try and figure out what's going on. Whereas if you're following them, again, while they're healthy, you can see what shifts, you have a better idea what's going on. Not perfect, but we take literally 135,000 measurements every time we measure someone.
Mhm.
So we're getting a much, much deeper profile. The analogy we like to use is that if your health is a 1,000 piece jigsaw puzzle, we're trying to get 600 or 700 pieces versus, say, 5 or 6, we would argue you get today. You
Yeah,
get very incomplete pictures.
yeah, and not only is it 5 or 6, but there's a pretty good chance that one or more of those mere 5 or 6 pieces aren't even relevant to what's going on.
Yeah, your heart rate in a doctor's office for most people is worthless
Why is that?
because they get nervous.
Yeah, yeah,
Heart rate jumps up 20 points.
yeah, yeah.
Whereas, believe it or not, if I were to pull your heart rate off of a smart watch, which is we do a lot with wearables as well, first thing in the morning, it's a pretty good indicator what your baseline heart rate is. And if you're stressed, either mentally or say physically, like you're coming down with something, your heart rate will jump up, and we can pick that up.
So there's a lot of directions we can go with this. I'm gonna start us on one track, and we'll just kind of see where we end up with this conversation.
Sure.
But obviously, something that's top of mind for a lot of people is diet and how diet affects metabolic health. Many different aspects of that we could discuss. What are some of the major questions in your lab that you've been addressing recently that have to do with the connection between diet, what you're eating, and what your metabolic health looks like.
Yeah, we're doing a lot in the area of glucose control, believe it or not, and diabetes and pre-diabetes more than that. So, because again, we're pretty keen on keeping people healthy. So this idea of prevention is, which is not so well exercised in today's healthcare system. Again, we do sick care. So it turns out, well, first of all, I'm diabetic. You may not know it if you were to see me in person. I'm a pretty skinny guy.
Hmm.
Yeah,
Type 1 or type 2?
a weird type 2. So
Really?
first of all, that's another thing. Diabetes is not just type 1 and type 2. It's probably more like type 1 through type 50. Uh,
Wow.
so type 2, we know we can subtype. For example, some people don't make insulin well. Other people don't release insulin from the pancreas. That's me,
Hmm.
actually. Most people, their cells don't respond to insulin.
I see.
Insulin lets your cells take up glucose. That's the most common form of diabetes, but there are many subtypes.
Yeah. So hold on. I just want to make sure I understand because I actually I didn't know that there was that much difference there. So type 2, broadly speaking, that means you have acquired insulin resistance and become diabetic late in life, as opposed to being getting it early in life because your pancreatic beta cells got destroyed. So
Correct. So
most,
that's type 1, by the way.
yeah,
What
that's type 1.
describe type
Type
1.
2, most commonly, people are producing insulin, but their cells are no longer responding to insulin as much. But you
Right.
have a different version.
That's correct. In fact, and it's not unheard of either. It's not common. So my version is that I do make insulin, my cells respond, but I don't release insulin from the pancreas.
Hmm.
And not only that, it turns out I don't respond to metformin, which is also kind of unusual.
And that would be a common drug that people do respond to.
Yep, and that's what most people do, and it works pretty well for most type 2 diabetics, and it's generally viewed as quite safe. These days, there's a whole bunch of other drugs out there you may know. These Ozempic derivatives, they mimic a hormone called incretin, and they're extremely effective. So anyway, I didn't respond to the most common drug. It's metformin, but I did respond to something that stimulates insulin release from the pancreas. It's called rapinolide.
Hmm.
And I actually worked well with that. Now, these days, I'm on actually one of these GLPs because they work even better for me. But yeah, so these Ozempic kinds of drugs. I
Yeah.
happen to be on one called Manjaro.
Yeah.
Anyway, they work incredibly well on me. But we think this is really important because diabetes has many subtypes, and the way you treat people very much depends on the subtype you are. That
Right.
is like I was just describing. I respond to certain drugs, but not others. The other thing we're discovering, here's an area of research that's very active for us, is we all respond to foods differently
Hmm.
from our glucose response. That is to say, some people, so first of all, backing up a little bit, here's the problem. 11.9% of people in the US are diabetic, 33% are pre-diabetic, and they're going to go on and become diabetic in their lifetime, the vast majority of those.
So roughly speaking, something like half-ish of adults are either diabetic or pre-diabetic in some way.
Right. It's totally out of control, and people don't take it as seriously as it should. This is an endemic that's quite serious because it leads to, heart failure, all these problems, increased
Yeah.
cancer later in life.
We often think of epidemics in the context of infectious disease, but we have an epidemic of this metabolic disorder.
We really do. Obesity and diabetes, and they're not identical, but they do go hand in hand. So most people are diabetic are overweight, but not all. There's a number of people like me who are thin diabetics,
Yeah.
probably because there's some subtypes different. So anyway, we're trying to figure out what the subtypes are and then how to better manage it. First through lifestyle, because we think that's probably a healthier way to do this. And one form of lifestyle is what you eat. I'm really a firm believer food is medicine. And I think more and more we're seeing this.
Yeah.
You may not realize this, but people eat 40,000 times more sugar now than they did at the end of World War II.
40,000.
40,000.
How do we how do we know that? What's the basic how
Oh,
do we
you just
gauge
looked
that?
at how much diet. There wasn't as
Yeah.
much sugar around now. Now sugar is in
Yeah.
everything.
When
A
and when you
bottle
say
of
when you
ketchup.
When you say sugar, do you mean like sort of the number of glucose molecules that end up in their body, or do you mean the sort of the mass of added sugars?
added sugars.
Okay.
Incredible. Yeah. It's true that people that had carbs, but it's a healthier form. So ultra processed foods, a good example, they're usually loaded with lots of sugar. And if you look at how much sugar is in a bottle of ketchup, it's most of the bottle.
Mhm.
It's incredible.
Was how do we get to that? Hold on, 40,000 times?
40,000.
But I'm a little perplexed here. So no one's eating 40,000 grams of added sugar per day, but people, 100 years ago were probably eating one or two.
Maybe not. As
Really?
straight up sugar, yeah.
Yeah.
Right at the end of World War II, probably not as much. Yeah, and just look at people's shape. If you look at diagrams of crowds, or not the photographs
Mhm.
of crowds from
Yeah.
people in the 1940s,
Yeah.
they don't look at all
Yeah.
like they look now.
So in essence, what you're saying is before World War II, there was little to no added sugars in things, but today it's kind of in everything.
It's in everything.
Yeah.
And kids, they get on sugar right from the get-go. Mine
Right.
did.
Yeah.
Yeah.
From in the womb, even.
Yeah. Yeah, that's true too. So anyway, we have a problem,
Mhm.
and so one is simply getting rid of that. Now, I did that initially. It's how I got when I first became diabetic. I'm a very interesting case, by the way. I'm genetically, I was predicted from my genome, my DNA sequence predicted my diabetes, and then, but I wasn't diabetic at the time. I only became diabetic after a very nasty viral infection. It's called respiratory syncytial virus.
When was this in your life? How old were you?
It would have been 2011, so I would have been, that's 14 years ago. So I guess I was 54.
Wow. So middle aged, and this was you had a genetic predisposition of some kind, but you needed this sort of environmental trigger for this disorder to manifest.
Correct. Yep. Yeah, and scientifically, it's very interesting. Obviously, I wish it hadn't happened, but scientifically, it turns out if you've heard of epigenetics, where you can get modifications on your DNA,
Mhm.
it turns out my metabolic genes got modified, and that's when my sugar went off. If you look at the kinetics of all this, meaning we've done very detailed analysis of my epigenetics. And you can see my sugar has gone off twice. When I first became diabetic, my methylation pattern shifted. It took about a year. I got it under control through lifestyle changes. I cut out all exogenous sugars. Yeah, and I was doing a lot of running and got my sugar levels down to normal. And then it actually came back after I stopped running. And I also got a
Huh.
second viral infection. So it's not 100% clear. I started running again, but I never got all the way down. I got it partially under control. But each time my glucose went off, my epigenetics change over a bunch of metabolic genes. It's
I
quite
see.
fascinating
So,
scientifically.
yeah. So in essence, it sounds like what happened is you got an infection. It sounded like a respiratory viral infection.
Yeah, that's right.
And then obviously your body reacts to this. There's an immune reaction. There's some kind of inflammation. There's stuff happening in your body. As a side effect of that, you had changes in DNA methylation. So different genes got turned on and turned off.
Right.
Some of those genes had to do with glucose regulation.
Yep, that's exactly right. And it's interesting now and that was the first time that had been shown where a viral infection, it's been suggested for type 1 previously, but not for type 2. And again, I'm a weird type 2. But now you may or may not realize that with COVID, somewhere between 2 to 4% of people become diabetic after a
Really?
COVID infection. Yeah, so it's probably a more general phenomenon than you think.
Yeah.
Quite a few people are getting something called chronic fatigue syndrome, if you've ever heard
Yes.
of
I've
that.
been reading about this.
Yeah, it turns out it's a lot more prevalent than people realize.
Right.
That's also often thought to be associated with the viral infection or some
Yeah.
other bacterial
Well, yeah.
infection.
Well,
Other-
did you see the new Yale study that just came out?
I did not, no.
So,
Oh, the one from Akio, yeah.
yes.
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 Yeah.
Basically there's a post-vaccination syndrome that you can see. And even, we're not going to dive into this, I don't think, but the punchline here is they were able to segment people based on whether or not they had 0 to 4 doses of one of the COVID vaccines, and also determine whether or not they ever had a previous SARS-CoV-2 infection, including whether, even if they were asymptomatic and didn't know it. And so, you had people that had a vaccine, that didn't have a vaccine, that had prior infection, that didn't have prior infection, 4
Uh-huh.
different cohorts there. They were able to show that people who had multiple doses of the vaccine, but no prior infection. Some of those people even had this post-vaccination syndrome that was probably
Mhm.
coming from the spike protein.
I see, but probably lower frequency than those who didn't get it. Is that right? Do you know?
I don't know about that. You saw it in both
We'll
groups.
have to
Yeah.
look it up. Yeah, we'll both
Yeah.
go back and
There
look at
was there was a small
it.
number of people who had spike protein in their blood almost 2 years later who had never had a prior infection.
That's showing up now more and more commonly. Yeah, this idea that this thing is lingering in some fashion.
Yeah.
And it's that in general for people who get long COVID, if they haven't been vaccinated, the probability of getting long COVID goes up. Um, so vaccination does, in general, seem to help protect.
Yeah. But I think what
It's
you're
not
saying
to say
is,
there aren't complications. There are.
well, yeah, I think the picture that seems to be emerging in my mind is so there's an interaction at two levels, at least. One, with the vaccines aren't as good as we would have hoped at preventing infection, but they do seem to prevent severe COVID, which would imply a lower overall spike protein load from an infection if you've gotten a vaccine. But at the same time, the mRNA vaccines, are engineered to have highly stabilized mRNAs that will produce a lot of spike protein potentially. And we're talking about people now who have gotten 2, 3, 4, even 5 or 6 doses of this, and there's probably something going on with the number of total exposures to spike, either from infection or from vaccination that people have had.
Yeah. Okay. Anyway, it's pretty clear. Yeah, people who get COVID, it does linger longer in some
Mhm.
cases. Yeah.
So anyways,
Yeah.
going back to your case, can you talk a little bit about like how when you first became diabetic, you've got this less common version of type 2 diabetes. How did, as you discovered this and as you started to get talking to your doctor about it, getting treated for it, how did that play out? I would imagine that maybe your physician probably wanted to start by treating you the way you would treat, the average type 2 diabetic, but then in your case, this probably wasn't the right thing to do.
Yeah, that's definitely the case. I mean, in my case, I caught it right away.
Mhm.
Now, I'm not saying everybody gets triggered that way. We
Yeah.
we we're studying this group of people, and I'd say most people just gradually get diabetic. But there's at least another person in our cohort who was like me, who spiked their way there as though something triggered it. So I think either way could happen. In my case, I just quickly changed my lifestyle because I saw it happening right away, and I gradually, as I say, got it under control, and that worked. And so I was pretty happy with that.
Mhm. How did you notice it was happening? What was
Well,
the,
because
the
I profile myself a lot. I'm
okay?
one
So
I'm one of my guinea pigs. So
I see.
we're studying this group of 110-ish people. We've been doing it now for well, for me, 15 years, and for this group, it's been about 12 years. We've been profiling them very, very deeply. And again, with the goal of trying to understand, it was mostly just can we use big data to better manage people's health? Can we use genome sequencing, for example, to better predict whether we can, to see if we can, again, manage your health? And in some cases, for example, from the genome sequencing, we found someone who had a heart problem. He didn't know but his genome said something was off, or he should have this heart defect, I should say. And it turns out he did. He and fairly young, didn't know it, and so he's on medication now. It was again another person was at high risk for cancer. They did a whole body MRI, and it turns out they actually had early thyroid cancer. So we're kind of bringing these technologies in to see if we can again better manage people's health. And in the case of genome sequencing, predict what they're at risk for and then keep an eye out for those things. In the case of these deep data monitoring from blood, see if we see something off. We caught someone with early lymphoma, for example, by the steep profiling, and two people with precancers. So we think this deep data really does give a better picture of your health and lets you catch things before symptoms. These people
Yeah.
did not have symptoms, and so that's the key. So back to my diabetes. I know I'm digressing a bit here, I guess. Anyway, yeah, when we ultimately, I couldn't quite get it under control just by lifestyle. I was changing my diet. I'd cut out all the sugars. I don't eat any of that stuff. I was being pretty active, meaning I was running about 45 minutes a run, say 4 times a week. It's about what my schedule could accommodate. And yet my glucose just gradually kept creeping up and up and up. So then I finally bit the bullet. I did try metformin, which is the thing you would normally take, and I didn't respond. I increased the dose. I still didn't respond. And then, believe it or not, I tried. So I hadn't figured out what my defect was yet, but I started lifting weights. Believe it or
Mm,
not, because the idea was that increasing your muscle mass is better for glucose homeostasis,
hmm.
and it worked in the sense I gained 10 pounds of muscle mass,
Wow.
but it didn't help my glucose at all.
Really?
Do a whole body MRI all the time too, so I could actually quantify all this and see my muscle mass increase. And yeah, it didn't help the glucose, and then it just kept creeping up and up and up. And then finally, through more measurements, I realized what was going on, that I was not releasing. I knew I was insulin sensitive,
Yeah.
and then I realized I wasn't releasing it from the
Yeah.
pancreas.
So you had non-insulin resistant type 2 diabetes. Your blood sugar is going up too much because you're not producing enough insulin.
Right. Well, I'm producing it. I'm just not releasing it from the
Oh, I
pancreas.
see. Okay, so the
So
cells
there's-
make it, but they're not releasing it.
It sounds like a subtle difference, but it makes a big difference from the drug standpoint, meaning
Yeah.
rapinolide is known to promote release from the pancreas,
Release.
and that works.
Yeah. You
Yeah.
don't need something that cranks up production because you already have production.
Correct.
Yeah.
Yeah. So it's interesting. And so this is why we think subtyping diabetes is a big deal, which is not done at all now. If we can do that. And it turns out, here's some studies we're doing now that's a lot of fun. Different people will spike their glucose to different foods. So they have these things called continuous glucose monitors. You may
Yep.
be familiar with those. So they measure your glucose every 5 minutes. And it turns out that our claim to fame, so to speak, is we started putting these on so-called normal people and pre-diabetics and discovered many of them have very bad glucose control. So obviously diabetics spike like mad after they eat something sugary. Turns out a lot of normal people do it, and many pre-diabetics spike pretty badly too. Some just as bad as diabetics, and they don't know it.
Mhm.
So, and it turns out different people spike to different foods. So some people, their glucose will spike to banana, others to pasta, some to white bread, some to brown bread, some to potatoes. We're all different. And Aaron Siegel, a different group, has shown that about 21% of that is due to your microbiome, the microbes in your gut, and
Mhm.
presumably how they metabolize fiber and sugar and such. But most of it's not explained. What we found is that for if you're insulin resistant, you'll spike to potatoes and pasta. So different foods have different response depending on your subtype.
Huh.
So we're trying to break this
I see.
down even
But is,
further.
is that a pretty robust finding that if you're insulin resistant, you will spike to those starchy foods?
Yes.
Okay.
um, it is. We all spike to white rice, by the way. White rice is very spiky. Yeah.
Okay.
And yeah, but so we think it's probably, this is just hypothesis, but we think it's the resistant starch in there that might be responsible. Now, how that interplays with the insulin resistance, we have some ideas, but it's not fully clear yet.
Yeah. Yeah.
So anyway, we ultimately want to, basically the end result out of all this is we want to build personalized diet programs.
Yep.
So if you know what spikes you, you avoid that and you eat the things that don't.
Right.
Okay. The idea is to push off then getting prediabetes and ideally diabetes for as long as possible. I don't know if you'll push it off forever, but you can try.
Hmm. So how often are you getting profiling?
Constantly. So I wear these monitors. So normally, two of my watches broke, but you can
Yeah.
see I have, well, here's a ring and two watches. Normally I have four. My hearing aids are sensors as well.
Yeah.
So at one level, I'm measuring my physiology all the time. At another level, I do these blood profiles and I wear a glucose monitor quite often. I'm not wearing one right now, but I do these omics profiling probably every 2 months or so. And if an
Okay.
adverse event like a viral infection, I take more samplings.
Yeah.
So I do a lot of this.
So all this stuff is very cool. I've done some of this to some extent myself. One thing that we need, and maybe if I'm lucky, you'll tell me we have it to some extent. You know, Is there any platform that can integrate the data from all of these devices so you can have it all lined up in one place?
Yeah, I should have started this program by saying I'm 100% conflicted because we spin off companies, and this what we're doing is academic. We're trying to
Yeah.
use these technologies to see what works, what's useful, what we can learn from them. But when things actually make sense, we'll spin this off as a company. So as an example, we have a continuous glucose monitoring company. It's called January Eye, and they build these personalized programs. They have a 50 some million food database. It's huge, and they can help you with all these predictions. Of course, they use AI for all this. So where I'm going with this, we have another company. It's a big data company. It's called QBio. They do whole body MRI and do a medical version of what I just said. They won't do the microbiome because we don't really know how to act on that yet, but they'll make all kinds of measurements. And right away, they've discovered, and they'll pull the data in and display it in a fashion that is so logical. To your point about integration,
Mhm.
they can bring in the MRI data and the molecular measurements and the genetics and display it in a way that says, all right, oh, look, these three things are off in your cardiovascular space. Keep an eye on that. Versus if you get your medical report today, they put the ions and things all together that don't necessarily make biological
Right.
sense, right? You
Right.
can't read it easily. You have to flip through the pages and try and figure it out for yourself. It's crazy. So basically, it's a matter of organizing the data in a very useful fashion. These days, of course, you can use AI to pull that information together and give specific recommendations. So we haven't talked about this yet, but we've set up another method for doing home microsampling and doing deep profiles on people, meaning you take little drops of blood. We used to do it from the fingertip. Now we do it from the shoulder. You mail it in, and we can measure 7,000 molecules from that drop of blood. And now I know what that sounds like, but ours actually works. Uh,
Yeah, yeah, yeah. No, this, yeah, for those listening, yeah, the technology has progressed since the infamous days of Elizabeth Holmes, and you can actually do blood work on relatively small amounts of blood and get lots of measurements done. I've done this myself. A
yeah.
lot, yeah.
Yeah. So anyway, we spun off a company there called Iolo that will measure 650 metabolites, pretty reproducibly.
What's that one called?
It's called Iolo, IOLLO. And yeah, and so you give the blood, you mail it in, and they give you back a report on these 20 wellness categories. Like they'll measure your oxidative stress, your inflammation, heart, and various things, and they even measure your biological age. Now, I know there's a lot of aging clocks out there,
Yep.
and so I think they're pretty good. Like the True Diagnostics methylation clock is
Hmm.
actually the methyl from Steve Horvath originally. I think
Hmm.
it's pretty good. But what's special about the metabolomics clock is it's actionable. And same with this information was telling you. So they'll break down, we call it ageotypes, your aging patterns. And so we can see your heart age and your kidney age and oxidative stress and these various things. And then you can use AI to basically take the information you give them about yourself, plus what's in the literature, and they make very specific recommendations. So it's not just exercise more, eat better. It's really like eat this exactly. You've been eating this already. We'll try this kind of thing. And so it works actually in the sense that people do improve their markers after they get the report back. And the hope, of course, is that that will lead to longer, healthier lives. That's the goal anyway.
Yeah. I want to talk about some recent work that you've done. And I guess what I want people to take away from this section is to have a better grasp on how to connect the dots all the way from what you eat to what's being produced in your body or by your microbiome to a change in metabolism via changes in gene expression. So linking, things like dietary fiber or other dietary components to your microbiome, to gene expression in your body and epigenetics, to metabolic change at the end. So you recently did a study that looked at something called short chain fatty acids and how these affect gene expression, how they're produced in the body. Let's start out really basic here. What are short chain fatty acids, and what are some of the major ones that people might hear about?
Yeah, they're these little molecules, acetate, propionate, and butyrate are probably the three most common ones. And so it turns out that they've been known for a long time to be healthy for you. They, boost your immune system, and exactly how they're healthy isn't fully understood, and certainly how they work is not understood. But it's known that fiber, everybody knows fiber is good for you. And we have a number of studies here, back to your earlier question about nutrition. We're actually trying to break this down because there are many different kinds of fibers. There are long chain, short chain, hydrophobic, hydrophilic, positive, negative. It's all over the map.
Mhm.
And so we've been testing different fibers.
But I
And,
guess with the unifying theme here for like what counts as fiber, it's something that we cannot digest on our own, and therefore microbes inside of us must break down for us.
Bingo. Yes. Yeah. Absolutely right. And so, and the fibers themselves, I mean, some of them have these compounds called polyphenols. You may have heard of them. They're getting more and more attention. They're antioxidants and probably have a lot of other health benefits too that people don't fully, know yet. But anyway, your microbiome breaks this down, and certain microbes then will actually produce short chain fatty acids as a consequence of all this. And so these short chain fatty acids are generally thought to be quite good for you. And there's plenty of evidence for that. So what people had been generally thinking is that there's an important modification on your histones, of things that package your DNA and affect gene expression. And basically, the prevailing view was that there's a very important modification. It's called acetylation. Uh, and these short chain fatty acids that mostly get produced by the fibers, they're called propionate and butyrate. They were thought to actually affect this other modification called acetylation.
Mm-hmm.
And I think what was kind of nice, what we did, and it stems from another lab's work, they showed that these things can actually show up on your chromatin itself, the packaging of your DNA. And I know this is getting pretty technical, but what we did show was that we actually went in and showed, yes, they're really showing up there. We actually saw exactly where they're sitting on your colon cells from, cells grown in a test tube in a petri dish. We could actually see exactly what genes were getting modified, if you will.
Hmm.
Their packaging was getting modified by these short chain fatty acids. So the idea is that we can make a direct link from fiber
Yeah.
to the actual genes that are getting affected. And it turns out it's genes important for gut integrity. They're these connections between your gut cells. They're called gap junctions and
Hmm.
such. And they're regulated by
Yeah.
these fiber modifications, these short chain fatty
I
acids.
see. So if I can summarize what you've just said so far, long story short, your gut needs to be have high integrity, meaning that the physical connections between the cells of the gut need to be in a good state. Let's just say that you don't want them too loose because then you'll have a leaky gut. Say you probably don't want them too tight anyway. You want them to be healthy, whatever exactly that means. That healthiness depends in part on the proteins they're making, which of course depends in which genes are being turned on and turned off and to what extent. What you just told us is that dietary fiber, you can eat fiber. Microbes, if they're in your gut, the right ones, will turn that fiber into short chain fatty acids. And these short chain fatty acids actually go in and affect gene expression. And some of the genes they affect are the ones that control the physical integrity of the gut.
You nailed it, and I appreciate you translating it for me. No,
So,
you're spot on. Yeah. So the bottom line is then fiber is making key molecules that are important for gut integrity. And as you point out, if you don't have that, you get a leaky gut, and then your immune system starts interacting. The microbes can get inside your bloodstream in some cases if you have a leaky gut. It's a big deal.
yeah.
So you really want a great intestinal. Both
Yep.
mucosal layer and having the cells, joined together.
So one question I want to ask you about this, and this is something I've been reading about, and my thinking is it has changed. I've now, my thinking has changed. I'll just leave it at that. Um, Something I often hear, and I want to get you to very carefully parse this for me. So it's unquestionably true that dietary fiber can get metabolized into short chain fatty acids that lead to good stuff for your health via the gut microbes that are in our gut. The question I have is Is that the only possible way to make these short chain fatty acids? And just to put all my cards on the table, it appears the answer to that is actually no. It's not the only way to get these things. What can you tell us about that?
Well, you can eat some of these things, believe it or not. You can take these as a supplement. I don't know this area so well myself, so it's not one I can probably talk about with any, informed state here. But
But one way to get these things is through fiber.
for sure.
Yeah.
And I think people like the concept of fiber. I certainly do because you do need again, for the mucosal layer of your intestine and just keeping that microbiome healthy. You may or may not know that 70% of your immune cells are in your gut.
Mhm.
And a lot of it's there to keep that balance between your immune system and the microbiome that's there, keep everything in check. And so that's a big deal. And you really want a healthy balance there. Um, and so, yeah, I think fiber is a great way to get it. As I mentioned before, it has certain fibers have these polyphenols, which are antioxidants, amongst other things, that are very valuable for your health as well. And so, yeah, your fiber is just, yeah, fiber is basically setting up a fermentation chamber in your colon that's just very, very valuable for your health. And so, sure, you could probably get some of this short chain fatty acid through diet, but it's not going to be quite the same.
Yeah, the work that I'm thinking of here, and there's I think very little of this work has actually been done, but I think his name is Peter Turnbaugh at UCSF, I believe. So he did a small trial in humans a number of years ago. They did a plant-based diet with fiber. They did an animal-based diet, which was basically just meat, eggs, and cheese. It had essentially little to no fiber. It was only about 5% carbohydrate. And what they showed is that in that animal-based diet, even though it lacked fiber, they nonetheless had butyrate and acetate production. It was somewhat lower than with the plant-based diet, but it was there. But then you had this increase in these other short chain fatty acids, isobutyrate, and some other ones. And
Right.
so it showed that, at least in principle, for some people, you can get production of you have a different profile of short chain fatty acids, but you still get them even in the absence of dietary fiber, which was interesting.
Yeah. And I don't know because we haven't studied those particular short chain fatty acids to what extent they wind up at the same gene targets
Yeah,
that the other ones. I don't know. That's a whole, this is really just the tip of the iceberg
yeah,
because there are a lot of these modifications and we have not pursued them all.
mhm,
But I think, for the first time, we'd like to think we've made some links
yeah,
between these short chain fatty acids and some
yeah,
pretty key genes that are important for our gut health.
yeah. But sort of in the abstract here, what you told us from these results, is you eat stuff, whatever that is, that stuff gets turned into other stuff, and then ultimately some of that stuff, in this case the short chain fatty acids, they literally go into the cells and affect which genes get switched on and switched off, which is going to determine the proteins that are present in that cell, which is going to determine basically the health of that cell and what it's doing.
Yeah, and I don't think it's just your gut either, because
Yeah,
these short chain fatty acids, you may know, they permeate your body.
yeah,
They'll go into your heart, they'll go into immune cells. So this is probably more systemic than just your gut. But we've kind of worked out at least some of the specific mechanisms for the gut, and I think it's going to be valuable to, map that out for the rest of the body as well. How do these things help your heart, for example?
yeah, yeah. And while we're on the gut, I want to get into some of the other results from that paper that you guys did. I believe there's a connection to colon cancer here. And what did you find there?
Yeah, we also looked at cancer cells, and it's true that some of the targets are these known oncogenes and things. And so, how that is affecting cancer, and those are known to promote cell proliferation. So, exactly how that part is working, I'm not as sure about, but we do know that they do change the gene expression and the targets do change in cancer cells. And to what extent then the fiber actually is making things worse in cancer, I don't know. But certainly for preventing cancer, I think we have pretty solid models that you're keeping good gut intestine, integrity, and I think that's valuable. For the cancer side, it's definitely true you're hitting another set of targets.
Mhm.
Some of them are involved in cell proliferation, which is what cancer is all about, as you know. If you start growing uncontrollably, you get cancer.
Mhm. And so some of the genes that these short chain fiber derived short chain fatty acids in this case affected had to do with the junctions between the cells of your intestines. So how they affect how tightly those cells are packed together, and therefore whether or not you're going to have a leaky gut and things like that. Any other types of genes that were families of genes that were affected?
Yeah,
Any
there were
patterns there?
ion channels and things that are again important for gut function. You need
Yeah.
bring your ions in, from the inside to the outside. So there were quite a few different systems there, but those were some of the most obvious ones.
Mhm.
The cell integrity and the transport genes were there were quite a few of those that we
Yeah.
saw that were targets.
And remind me, in this paper, you were looking at, were you just looking at rodents or were you looking at human cells in vitro as
We
well?
did both.
Okay.
We did it initially in human cells in vitro, and that told us what the targets were. It's very easy to map them out. And we also did human cancer cells in
Mhm.
vitro, and then we followed it up we basically had high fiber diets for mice and showed similar kinds of targets in the gut in the mouse. Were presumably hit by these short chain fatty acids. Not presumably, they were. So the bottom line is that that's nicer because it's a more controlled system, and we could show then that it was going on in a living organism.
Yeah, yeah. So at least directionally, similar effects in the cultured human cells and in the mice.
Correct. Yeah.
The other thing, among many things that you work on, the other stuff that I thought was interesting is this concept of the lipidome and looking how the fatty acid profiles of the body change in health, in disease, in aging. Can you start to talk about that a little bit? Give us a sense for what the lipidome is and how big and how diverse it is.
Well, it's huge. I mean, we don't even know all the lipid molecules out there because you can get many permutations of lipids. So lipids, people just thinking, oh, they're greasy molecules. And they've always had kind of a bad reputation, I guess, because of that connotation. But they're critical for your cell integrity. They make up your membranes are lipids, and obviously there are just tons of those. But it turns out they're very important signaling molecules too, sphingolipids, ceramides, things like this. And it's an almost infinite number of molecules, it seems, because you can get all these different permutations depending. Again, this gets into chemistry, but whether the bonds they're called saturated versus unsaturated, which,
Yeah.
by the way, is a big deal. But anyway, so we don't know what most lipids do beyond, the integrity stuff in the cell, and especially how they signal. So we just started profiling them. We know they're important for energy metabolism. For example, when you run, yes, you use up your glucose right away, but then you start using your fatty acids for
Mhm.
metabolic purposes, carnitines and things like this that get derived from these. So anyway, so we just started profiling them through health and disease just to see what shifts are there. And so we're getting some initial observations about, again, what some of these things might do. So we sort of associated some classes of lipids, for example, as pro-inflammatory, others as anti-inflammatory, in addition to ones that are already known. There's some characterized things out there already known to be important for inflammation, for example. But we found new sets that I think are going to be useful to know about. No, they could wind up being important markers of disease then.
Yeah, yeah. I believe in that paper you looked at things like the transition into insulin resistance and other disordered states. Can you talk about that a little bit? How some of these, going from a state of health to unhealth or vice versa, how did some of those transitions look in terms of how the lipid profile?
Full
version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to take just a minute to tell you about an app I am partnering with called Readwise. Readwise is an app that organizes and helps you get the most out of your digital highlights. I use it to organize all the highlights I make in my digital books on my Kindle, and so if you're like me and you make a lot of highlights and you like to revisit them often to refresh your memory, Readwise is the perfect app. You can also take photos of any physical books you've highlighted and upload those. It also has cool features that allow you to share your favorite highlights and quotes from books on social media, and it syncs with note-taking apps like Evernote, Notion, and Roam. You can tag, search, and organize your notes and highlights on Readwise, and it helps you connect ideas in new ways and retain more of what you read. So if you click the link in the episode description, you can get Readwise for free for 2 months when you sign up for their annual plan. That plan is only $7.99 per month, and it's a relatively new app, so they're adding new features often, and if you sign up for the annual plan today, you can lock in that price, which will stay at $7.99 even if the price increases in the future as they add more. So if you do a lot of highlighting and note-taking and you want a good way to organize all that information, check out the